It is rare that you can spend maybe 20 million. It sounds a lot, but in drug development terms, it's just a drop in the bucket. To spend 20 million and in 1 to 2 years, essentially have all the answers that you need to determine whether you have what would then appear to be an enormously valuable asset. The questions that we need to answer are twofold. With the monoclonal antibody, with KappaMab, we need to see whether by raising the dose, which has increased efficacy with similar antibodies that have already gone into development, we can see even greater enhanced efficacy. And we can do that in about 30 patients, we estimate, which in the grand scheme of drug development is quick and cheap process to get an answer that we anticipate having sometime in 2024. The other question we would like to answer is, will the CAR T data that's been very positive in mice, will that be reproduced in people? And the Peter Mac Institute is already planning to actually give a properly manufactured CAR T to a group of patients. And if our CAR T is anything like other CAR Ts, 6 to 12 patients is enough to see whether you're in the efficacy zone or not. So that's another 2024, likely 2024 event. If we are active, if we have an active and effective CAR T, that will become an effective therapeutic. We know that from past experience. It almost certainly validates that a bispecific antibody, which sort of does the same thing in a different way as the CAR T, is highly likely to be effective as well. And obviously we will have the dispositive data on the antibody. And at that point, it's all downhill. It's just a matter of taking the time and spending the money to fulfill regulatory requirements. That does take time, it does take money, but you've written down almost all of the risk. So in drug development terms, this is almost a sort of perfect case that we have. It is rare that you can spend maybe 20 million. It sounds a lot, but in drug development terms, it's just a drop in the bucket. To spend 20 million and in 1 to 2 years, essentially have all the answers that you need to determine whether you have what would then appear to be an enormously valuable asset. When I first joined the industry, the head of R&D at Glaxo at the time said the aim of drug development is to take the shortest route to the negative answer. Get an answer as quick and as cheap as you can. And that's not always possible. The checkpoint inhibitors that were in the balance for 7 years, and they were finally shown to work after a long, expensive 5-year randomized placebo-controlled study in 750 patients. We had to wait that long to even know that we had an answer. That's completely different with HaemaLogiX. It's quick, and in the grand scheme of drug development, it's dirt cheap. There are some dispositive studies in drug development which are blinded. No one knows what's going on. No one knows whether they're getting the active or the placebo treatment, and you have to wait sometimes years to the end of that study before you actually know the results. These studies are different in that they are open label. We know what's happening with every patient as we go through the study. for both the monoclonal and the CAR-T. So if we are, and it's possible that we could see interesting activity early on. That being said, each study does have an element of starting at a somewhat lower dose and escalating the dose. So you have to kind of probably wait until that happens, but that's a smaller part. So yes, there will be opportunity to see some early data, but this does have the advantage from the point of view of all of us investigating the drug of having more or less a real-time evolving readout, which again encourages making sure you have, if it's looking good, that you have lead time to get the extra resources to exploit success.